In this video, we're going to learn about the structure of DNA, the molecule we'll be discussing all year in this genetics class. The key idea for this unit is that the structure of DNA determines its function. DNA has two important jobs. One, it stores information. The genes in the DNA molecules contain the code for making proteins. They do this through the processes of transcription and translation. The second semester of this course is going to focus on gene expression and how we go from the code in DNA to proteins. Our first semester will focus on the second job. DNA transmits information. What that means is DNA can make copies of itself, and those identical copies can be passed on to new cells. Now, this is really important for the growth and reproduction of organisms. So let's examine the structure of DNA more closely to determine how its structure allows it to do those two jobs, make proteins and copy itself. Well, the first thing we should know is what type of organic compound is DNA. DNA, N-A, stands for nucleic acid, okay? And a nucleic acid is a polymer or a chain of nucleotides, okay? So this is a DNA molecule here. You probably remember from biology that a DNA molecule is typically a double helix. So that spiral shape in this case has been flattened out. Now let's circle some nucleotides so you can see how these nucleotides are joined together. Well, a DNA molecule actually has two strands of nucleotides. So I'm going to essentially draw a line down the middle. This is one strand on the left and one strand on the right. Each nucleotide has three parts. So there's one nucleotide. The next nucleotide is bound below it. And then the next one after that. And after that. So you can see that each side has four nucleotides. The other side has four nucleotides as well. They're just pointing in the opposite direction. More on that later in this video. So DNA is a nucleic acid, a polymer or chain of nucleotides. Two chains put together, to be exact. DNA stands for deoxyribonucleic acid. Okay, so that's a big long word, deoxyribonucleic. But the deoxyribo comes from the sugar deoxyribose, and that is the sugar that is found in nucleotides. So I am going to circle the sugars in red. So each of those little blue pentagons is the sugar deoxyribose. So let's examine a nucleotide structure in more detail. I circled a bunch of them on the DNA molecule on the page before. So each nucleotide, okay, has three parts. The first one is a phosphate or a phosphate group, whichever one you want to call it. That is this right here. Okay, a phosphate group has a phosphorus in the center surrounded by oxygens. And those oxygens, although it's tiny and it's harder to see, those oxygens have negative charges. So a DNA molecule, because it has so many phosphate groups, is a negatively charged molecule. And that'll come up a few times throughout the year. So, phosphate group. The other, and I'll put a red star. So you see I circled that with red. Okay, in this case, my deoxyribose sugar, I'm going to do in green. Okay, this is my deoxyribose sugar right there. It's also called a 5-carbon sugar because there are 5 carbons that make it up, or sometimes it's called a pentose sugar. So pent, like pentagon, is 5 sides. So what's not shown is that each corner here and then one up here is a carbon. So that's 5 carbons that make it up. Now, nucleotides come in two different types. I keep talking about deoxyribose, and that is the sugar in DNA. There is another nucleic acid called ribonucleic acid, or RNA, and it uses the sugar ribose. We'll learn lots about RNA during our second semester when we learn about gene expression. The last part of a nucleotide structure is the nitrogen base, which I'm going to do in blue. So this base, or nitrogen base, is what I'm going to circle up here. Do you guys see all these N's? Okay, those are the nitrogen. So like sometimes it's a nitrogen base, sometimes it's just a base, sometimes it's called a nitrogenous base. All of those are fine. There are 5 different nitrogenous bases. Okay, now they come in different shapes. A purine are nitrogen bases that have 2 rings, whereas pyrimidines have 1 ring. Now, let me show you what I mean by that. Okay, so guanine and adenine. So here's adenine right here, and here's guanine. Do you guys see how they have 1, 2 rings? Okay, and then this guy's got like a pentagon attached to a hexagon. So that is a purine. They have 2 rings. Okay, I don't necessarily need you to memorize what a purine is, but it does come up again when I talk about the structure later on. So it's important to know that. Adenine always pairs with thymine, which is T, and you'll notice it has 1 ring. It is just a hexagon. Same thing with cytosine. What I didn't draw on here or include picture of was uracil. Uracil is RNA only, so it's only found in RNA. Same thing with thymine. Thymine is DNA only, so these are unique bases depending on the type of nucleic acid. Now, DNA nucleotides have the same basic structure. They all contain a phosphate group and a 5-carbon sugar. They only differ in their nitrogen bases. So you can have adenine containing nucleotides, thymine containing nucleotides, guanine containing nucleotides, and so on. All genes are made up of stretches of these 4 bases, arranged in different ways and in different lengths. So the orders of ATCs and Gs in DNA, that is what determines the genetic code. Here's another picture you can see. This is a polynucleotide. Let's start on the right. So here you can see nucleotide, the little round phosphate group, the green pentagon sugar, and then the yellow base in this case. So notice this base, because it's got 2 rings, is a purine. So it could be either adenine or guanine. In my next nucleotide, I only have 1 ring, and so that could be a pyrimidine. Cytosine or thymine if it's DNA. If this polynucleotide was RNA, then it could be uracil. Okay, so you can zoom in on each of those. In this case, this polynucleotide has 1, 2, 3, 4, 5, 6 different nucleotides making it up, and they all have the same basic structure: phosphate group, that doesn't vary, a pentose sugar or 5-carbon sugar, deoxyribose in DNA or ribose in RNA, and then a base. Okay, so hopefully that gives you a better idea of nucleotides and how they work. Now let's talk about how those nucleotides go on to form the double helix. So a DNA molecule is made of 2 strands of nucleotides twisted into a double helix. So this over here is the double helix. So you can really see, let me just highlight. So here is one side of the DNA molecule. So that's 1 polynucleotide, 1 nucleotide strand. And then here is the other side. So we got 2 strands that are twisted up. And it looks almost like a spiral staircase or a twisted ladder. Okay, what is making up the red and blue parts that I made? So this would be like our blue side, and then this would be like our red side, okay? What's making up those outer kind of ribbons, the railings of our staircase or the rungs of the handles of our ladder, things like that? That is what we call the sugar phosphate backbone, okay? So the sugar phosphate backbone is what's in this gray box. And you'll notice that it goes, in this case, phosphate, sugar. And so it just keeps repeating that over and over again, that pattern, and that is why it is called the backbone. Now, notice the other side, it starts with sugar, then phosphate. We'll talk more about that later on, but basically it's still a sugar phosphate backbone. It's just running, one is running, say, up, and the other side is running down. We call that anti-parallel. More on that to come. Okay, so the sugars and the phosphate groups are bonded together with covalent bonds. So basically all of these dark black lines, those represent strong covalent bonds. And that's important because we want this backbone to stay together. Now, in order to get to the information, the cell has to access the bases, the A's, the T's, the G's, and the C's. And in both copying DNA and making proteins, the DNA molecule essentially has to unzip down the middle. Now, this backbone needs to have strong bonds. So as the DNA molecule unzips, the sides stay together. We don't want the DNA molecule falling apart when we pull it apart down the middle, because if we lose the connections between our nucleotides, we lose our code. The A, T, C's, and G's, in order, that code that determines proteins, it's all gone. So we need these strong outer rails to be held together. The inside, on the other hand, are paired nitrogen bases. So you can see A pairs with T and G pairs with C. Now notice these bases are tucked on the inside, so it's basically these right here. You can see like all these the paired bases. They're on the inside of our double helix. And they're held together with a weak bond. The type of bond that holds them together is called a hydrogen bond. Hydrogen bonds take much less energy to break than these strong covalent bonds over here. So notice they're not even a solid line. They're represented as a dashed line. That's supposed to be weak. And that's important because I mentioned on the last slide that we have to unzip our DNA molecules. We have to basically separate them right down the middle in order to get access to the A, T, C's, and G's, the bases that have the code that we need for protein synthesis or DNA replication. So both jobs need the DNA to separate. As a result, it's important that we have these weak bonds. Now, you'll notice that A and T are always paired together, whereas G and C, we'll do these, are always paired together. And that has to do with the number of bonds that they can form. So you'll see that, let me erase some of this so it's a little clearer. But you guys see how the dashed lines in between an A and T, there's always 1, 2 hydrogen bonds. Okay, you can see them right here, 1, 2, 1, 2. But in a G and a C pair, I'm going to use a different color, there's 1, 2, 3. So there's always 3 hydrogen bonds between G's and C's. That is why you always have A and T pairing and G and C pairing, because they make the right number of bonds to want to stick together. So there are a few, in addition to the basic structure of the double helix, there are a few qualities that are going to be important and we're going to bring up several times throughout the year. Okay, the first thing is the idea that the DNA strands are complementary. That means they basically match up. They're not identical, but they complement each other. So we have pairs of bases that always go together. A pairs with T, adenine pairs with thymine. And G pairs with C, guanine pairs with cytosine. So if you have, for example, a T on one side, you would have an A on the other. A C would pair with a G. Pause the movie right now and see if you can fill in the rest of the complementary pairs of bases. All right, we're back. So let's fill in the rest. If you have an A, that's going to pair with a T. T with an A, G with a C, A with a T, G with a C, and C with a G. You notice how we talked about the fact that A's and G's are purines. They have 2 rings. Do you guys see how they're a little longer? They show that they're a little bigger. Whereas T's and C's are pyrimidines and they're shorter. Okay? So that would be the bases that would go on the other side. They complement each other. The cell can basically read the bases chemically through enzymes and know if there's a T, the A should go opposite and they should fit together with the correct number of hydrogen bonds. Now, because of the fact that we have A and T and G and C, hopefully you noticed that a purine always bonds to a pyrimidine, right? We have a double ring purine, okay, like this column right here, and a single ring pyrimidine. And so you always have 1, 2, 3 rings across, okay? So what that means is the DNA molecule has this nice equal width. It's always got 3 rings across, and so you have this kind of like smooth molecule. Now that's important because when we learn about mutations, if the wrong nucleotide is put in, so let's say we have an A and it's supposed to be paired with a T, okay? But something goes wrong and instead of a T, the enzymes in the cell put in a G. A and G don't pair. In fact, that would be 2 rings here and 2 rings here. That would be 4 rings across, not 3. Your DNA molecule would essentially have a bump in it, okay? And so the cell actually can pick up on this. There are enzymes that track down these mismatched pairs because of these bumps and will cut them out and basically cut them out and it'll put in the correct nucleotides so you get the nice equal width. So this equal width thing comes into play when we talk about DNA repair and mutations. There are some other dimensions to notice that DNA molecules always have, so this consistent width throughout. Also, you'll notice that the bases are spaced out, so they have an equal distance between the bases of 0.34 nanometers. The helix turns every 3.4 nanometers, and every turn there are 10 layers of bases. So basically they'd be like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, in every turn. So those are just some consistencies we see no matter DNA from a bat or a turnip or a human. We see these consistent dimensions of molecules, DNA molecules. All right, and the last quality of a double helix. Okay, I've mentioned this in this video before, but the strands are something called anti-parallel. Okay, you probably learned back in geometry that parallel means that the sides run side by side but don't intersect. Okay, so that's what happens in your nucleotides. You have one side and then you have another side and they don't intersect, but they run in opposite directions. and that's the anti part. One points up and the other points down. Okay? So what exactly do I mean? Well, in biochemistry, the DNA molecules, we say one runs 5' to 3', while the other runs 3' to 5'. What in the world does that mean? All right, let's look over at this over here. This is deoxyribose. Okay, so I'm just going to say deoxy. All right, and it is a 5-carbon sugar. 1, 2, 3, 4, and then 5. Oops, 5. All right, 5 carbons. Now, each of those carbons are numbered. So if we were to go around this sugar ring like a clock, it'd be 1, 2, 3, 4, 5. Okay. Now, the 3s and the 5s are referring to which sugar? So, the 5' end, this 5-carbon sugar over here, is always where the phosphate groups are attached. So, attached onto this OH, you would get your P. Okay, this would be your phosphate group that's attached there. The 3' end is always the bottom of the sugar. So 1, 2, 3. So this flat part of the sugar is always the 3' end. So here you can see it over here. So here's our phosphate group. That's our 5' end. Here's our bottom of our sugar. That's our 3' end. Okay. Now if you look on the other side, oh, this molecule's got the sugar here, whereas over on the other side, there's a phosphate group. So they're opposite each other. And then here, down here is our phosphate group. That's our 5' end. Okay? So that's what we mean by 5' and 3'. Think of it like pencils. Okay? You have a sharpened pencil. One points up and the other points down. Okay? They're running opposite each other. All right. So here is a DNA molecule. Let's see if we can figure out which ends are the 5' and the 3' end. Let me give you a hint. 5' is your P. The 3' end is your sugar. Specifically, you're looking for something called the hydroxyl group, the OH at the bottom of the sugar. That's what will tell you the 3' end. So pause here. Look at this molecule. It's kind of, I had to flip it horizontally to fit it on the slide, but you can still get the same idea. Think about which end, the left or the right, is the 5' and 3' for the top part and the bottom part. So remember, this is one side of your DNA molecule, and here is the other. All right, pause, and then we'll come back. All right, so hopefully you figured this out. Let's start over here on the right. On our top strand, phosphate group is 5', which means down here, this is our sugar end, this is our 3'. Okay, so this half of our DNA molecule is running this way. Okay, now if we look at the other side, it should be the opposite. This should be the 5', because we have 3' over here, so this should be our 5' end. And what do you know? We got a phosphate group, so that's our 5' end, which means down here is our 3' end, our sugar with the hydroxyl, the OH. And notice it's opposite of it, so this one down here is running the opposite direction. Okay, and that'll come up because when we talk about how DNA gets copied, or we talk about how DNA gets read by enzymes, they like to work in one direction. We'll often say this enzyme reads it in the 3 to 5 direction and builds in the 5' to 3' direction. So understanding this now will help us when we understand the technical aspects a little bit later on. So hopefully you have a better understanding of how the structure of DNA is set up, and that'll help you build on that knowledge throughout the year when we talk about the functions of DNA.